Hello, in this video we will walk through the processes of DNA replication, transcription, and translation, showing how genetic information flows in cells. DNA nucleotides consist of a deoxyribose sugar, phosphate group, and a nitrogenous base: adenine, thymine, cytosine, and guanine. DNA replication begins in the nucleus of eukaryotic cells and the cytoplasm of prokaryotic cells. For this model, we'll focus on eukaryotic cells and the nucleus. First, the enzyme helicase winds the double-stranded DNA, breaking the hydrogen bonds between complementary bases, adenine with thymine and cytosine with guanine, and this creates a replication fork. Topoisomerase comes in to prevent the DNA from supercoiling, while primase lays down RNA primers. These primers allow DNA polymerase to start adding DNA nucleotides. DNA polymerase works in the 5' to 3' direction, meaning that it adds nucleotides to the leading strand continuously. However, on the lagging strand, DNA is synthesized in short segments called Okazaki fragments, which are later joined by DNA ligase.
Next, let's talk about transcription, which occurs in the nucleus for eukaryotes and the cytoplasm for prokaryotes. During transcription, DNA is transcribed into messenger RNA, also known as mRNA. First, helicase unwinds the DNA, then the RNA polymerase binds to the promoter region of the gene and starts synthesizing RNA. RNA nucleotides consist of ribose, phosphate groups, and nitrogenous bases. Adenine pairs with uracil, and cytosine pairs with guanine. RNA polymerase adds RNA nucleotides complementary to the DNA template strand, creating a strand of pre-mRNA. Once transcription is complete, the pre-mRNA undergoes modifications such as the addition of a 5' cap, poly, A tail, and splicing to remove introns. This process results in a mature mRNA strand that can exit the nucleus and enter the cytoplasm for the next stage, which is translation.
Translation takes place in the cytoplasm in both prokaryotic and eukaryotic cells, specifically at the ribosome, which is made of ribosomal RNA, or rRNA for short. This process begins when the mRNA binds to the small subunit of the ribosome. In the initiation phase, the start codon, usually AUG, is recognized by transfer RNA, also known as tRNA, which carries the amino acid methionine. In the elongation phase, tRNAs bring amino acids to the ribosome. Each tRNA has an anticodon that pairs with the codon on the mRNA. As the ribosome moves along the mRNA, tRNAs continue to bring amino acids, forming a growing polypeptide chain. Finally, when the ribosome reaches the stop codon, the termination phase begins. The completed polypeptide is released, and the ribosome disassembles. This polypeptide will fold into its final protein structure and carry out its specific function within the cell.
There are multiple types of mutations that can alter DNA, 3 of which are a point mutation, which changes a single base pair, or insertions, which occur when one or more nucleotides are added to a DNA segment, and finally, deletion, which is a type of DNA mutation that occurs when a part of a DNA molecule is lost during DNA replication.